BioCentury this week is brought to you by Evotec. Evotec is a life science company pioneering drug discovery and development by integrating deep disease expertise, AI-driven innovation, and advanced technologies. Working across modalities, Evotec's fully integrated R&D value chain and flexible partnering models accelerate the journey from concept to cure. A key area of focus is cell therapy, where Evotec leverages cutting-edge iPSC technology, manufacturing know-how, and translational capabilities to deliver scalable, off-the-shelf solutions for complex diseases. From discovery through GMP production, Evotec supports the development of cell-based therapies, advancing regenerative medicine and bringing transformative treatments closer to patients.
The latest innovations in shuttling therapies across the blood-brain barrier, plus a conversation with GSK's CSO, Tony Wood, and the new FDA Commissioner's National Priority Review Vouchers. We'll be discussing all of this on today's BioCentury This Week. I'm Stephen Hansen, one of the directors of Biopharma Intelligence here at BioCentury, and joining me are my colleagues,
Selena Koch, Executive Editor.
Lauren Martz, Executive Director of Biopharma Intelligence.
And Steve Usden, Washington Editor.
Great, and so I am filling in for Jeff this week, who has been whisked away, along with Simone, over to Shanghai for our 12th annual China Healthcare Summit, which is brought to you by BioCentury and BayHelix. This year's conference, which starts on Wednesday, will be discussing China's innovation moment. We've already done a preview show that you can check out, and Jeff will be wrapping up the summit on Friday with a live podcast from the event. So keep your eye out for that. But we will dive straight into what we've got for you this week. So I think we'll start with Selena. Technologies for transporting molecules across the blood-brain barrier kind of seem to be all the rage now. Maybe before the end of the show, we could see whether any of us can say blood-brain barrier 3 times fast. I'm not sure if I can do that, but maybe I can get you to do that, Selena. But in the meantime, maybe you can tell us a bit more about these shuttles.
Blood-brain barrier, blood-brain barrier, blood-brain barrier. There you go.
Very good.
Well, I think we can
Very
go BBB. I think it's all right. I think starting with the magnitude of the problem these shuttle technologies are trying to solve would be good, because it really struck me when I was thinking about this story. There are a few problems that are wider reaching or longer standing when it comes to CNS drug development. The blood-brain barrier not only keeps out many small molecules in its job, which is a critical one for life, but it almost entirely excludes antibodies. So, I would encourage listeners to think about what it would be like to treat cancer or autoimmune diseases without the use of antibody therapies, right? These have been standard modalities alongside small molecules in the industry for a very long time now. But in the case of brain diseases, they've made virtually zero impact. That may be starting to change with the approved anti-amyloid antibodies, but, we will see. So typically, when you systemically infuse an antibody, less than 1% makes it into the brain, and often far less than that, less than 0.01%. And another issue is when it gets in the brain, where it distributes, because the way it's getting in is not active transport, it's leak in small quantities. You see a little bit of antibody present in the superficial, the most superficial layers of the cerebral cortex on the outside of the brain and right around the major vessels. And that's about it. So you're getting neither deep nor broad distribution, which makes it very hard to treat diseases with antibodies or protein therapies of any kind. And when it comes to RNA therapies, so antisense and siRNAs, these have obviously been proving their worth in many diseases in recent years, but those have to be delivered into the cerebral spinal fluid or even directly injected into the brain tissue itself, which we all know is inside of our skulls, right? So these kinds of invasive delivery methods, they can be workable, and they are workable in some cases, but I think it's fair to say they're not ideal. They're not scalable. And so they're reserved for the severest diseases, right? So if you could solve this problem, you could not only treat more patients with the severest neurodegenerative diseases, you could even think about psychiatry with biologics.
Selena, can you just kind of walk us through the basic mechanisms that are being pursued here? Because I know that there have sort of been some older, sort of some of maybe the first generation technologies that were developed to kind of try and surmount this problem. And then I think those, we've got some next generations that are coming along. So maybe can you just walk us through kind of the biology here?
Yeah. So the most widely used and most advanced receptor on the endothelial cells that make up the blood-brain barrier is transferrin. That's an iron transporting system that companies have co-opted in a Trojan horse-like way to smuggle their biologic into the brain. And I think in the early days of this, we saw some troubles that gave rise to kind of principles now, things that need to be solved and to get state-of-the-art kind of tech. So none of these blood-brain barrier receptors that people are targeting, well, I shouldn't say none, but who knows, close to none at least, are only expressed in the brain endothelium, right? So you need to avoid loss of drug in the periphery. You need to avoid peripheral tox. And so with transferrin, you can see that reticulocytes express a lot of it. So the early versions of these shuttles caused anemia, and you lost some drug that way as well. So companies have taken to, targeting epitopes, conformations that are more specific to the brain. That's one of the tricks. Another is the affinity of the binding. You can play around with that. So higher affinity binding is actually not better. You tend to get the drug and receptor complex shuttled toward degradation pathways within the endothelial cells. So basically the drug has to bind the cell, has to be internalized, has to traffic across the cell, has to be released into the brain tissue on the other side. And if it gets stuck inside the endothelial cell, then you fail at that step. Another way you can get stuck at that level is if you have multivalent binding, which can cross-link all the transferrin receptors on the cell membrane, which tends to also prevent your drug from getting in the brain. So low affinity or pH-dependent affinity that in the endosomal compartment where it traffics allows the drug to be, which has bound the receptor, then to be released from the receptor so it can be released into the brain. That can also work.
So Selena, what you mentioned how little of the drug, is sort of naturally able to cross the blood-brain barrier. What sort of efficiency gains are we seeing now? Maybe even relative to that first generation technology. Like how much better are these working now? And do we have some clinical proof of concept that points us to the fact that we're actually seeing drug getting into hitting its targets in the brain?
Yeah, we are. We're sending some really interesting demonstration projects. Well, first I want to say that the level of activity across the companies that I've been talking to in the last few years suggests that there's widespread research onto this. Like every farm is working on it, lots of biotechs, academics, there's lots of deal activity, so on and so forth. So there might just be a critical mass emerging to where this is going to be solved. And it's a question of when and by, how. But there's already been some demonstrations that it's working. So Denali Therapeutics was a pioneer in this space. They remain so, and they have a drug under review for a rare lysosomal storage disorder, Hunter syndrome, with a PDUFA date in January. So we could get our first US approval of a shuttle-enabled enzyme replacement therapy soon. There's already one of these for Hunter syndrome approved in Japan by a small company called JCR Pharmaceuticals. So that's the lead. And that makes sense. Because even if you make it through all those steps I just laid out, you then have to find your target in the brain. And so if you're going intracellular, generally you're going in through endocytosis, and you end up in the lysosomal compartment. So treating, if that's where you want to treat in the lysosome, that makes a lot of sense. So enzyme replacement therapies for lysosomal disorders is kind of a first area. The other is antibodies for extracellular targets. And so here we see the proof case of anti-amyloid antibodies for Alzheimer's. And I think this is actually a pretty good application to show the power of these technologies. So Roche is a leader here, and it's produced phase 2 data that are pretty compelling. So both on the efficacy side and the safety side of that efficacy safety profile anti-amyloid antibodies have some challenges, right? One of them is the risk of brain swelling and brain bleeding. And that kind of comes down to where the antibodies distribute when they get into the brain, because this risk is much more pronounced in people who have something called cerebral amyloid angiopathy, which means they have amyloid deposits, not just in the brain tissue, but kind of in the walls of the blood vessels. So if that's the only place you're sending your drug, is right around these major vessels, then you're causing inflammatory reactions right there that can break down the integrity of the vessel wall. You can get edema, you get hemorrhage. And what Roche has shown so far is that when they link their anti-amyloid mAb to a blood-brain barrier shuttle, it distributes much more broadly and deeply through the capillary beds. And a lot of it can avoid the cerebral amyloid angiopathy, and the rates of brain swelling and bleeding have been far less than would have been. It's not zero, right? It doesn't fix the problem entirely, but it's far less than what we've seen with the naked antibodies. So that's great. And then you're getting more drug in the place where it actually needs to be effective. So far we've seen that translate to clearing amyloid plaques a lot faster, deeper.
Right. So,
with lower doses.
So the theory is that you can potentially get better efficacy and avoid some of those safety issues that the naked antibodies have seen so far. Yeah. Well,
those are the leaders. There are
Great.
others. If you're interested in the deck that we published, there's pipeline charts for 3 major areas: lysosomal disorders, extracellular targets such as amyloid and others, and then RNA-based oligos for intracellular targets. Those are a little further behind in terms of like disclosed programs and data. Many companies are working on that. What that promises is to be able to access intracellular targets, but it's complicated there too, because once you've got into the endothelial cell, across the endothelial cell, out of the endothelial cell, into the brain, and you got to get taken up by the target cell, the neuron or the glia, you're once again in that endosomal compartment. Now you have to get out of the endosome to access to your intracellular target. So it's like yet another membrane crossing, right? And nobody's solved that problem for oligos in any system, let alone the CNS. But we have seen them be effective through basically they get sequestered in the endosomes and they kind of leak slowly. And so if the conditions are right, you can get this time-dependent release that actually can work in your advantage sometimes, because it can make RNA modalities, which are notoriously unstable and get degraded quickly in the body, last for weeks or months. But it's yet another factor to consider, the engineering and the product profile.
Yeah. Well, very cool. Well, everyone should check out Selena's story. It's up on the website. It's a really cool deck showing you sort of all the mechanisms and the pipelines, as she said. So check that out for sure. We'll take a quick break and be right back.
BioCentury This Week is brought to you by the 12th China Healthcare Summit in Shanghai. From in-licensing to new co's, cross-border deals from Asia are transforming global biopharma. Meet top decision makers and investors at the 12th China Healthcare Summit to explore emerging opportunities and tackle key challenges in this rapidly evolving landscape. Join us in Shanghai October 22nd to 24th to get a first-hand look at China's dynamic life sciences ecosystem. Register and learn more at biocenturychinasummit.com.
Welcome back. We'll now turn to Lauren. You had an interesting conversation with the Chief Scientific Officer of GSK, Tony Wood. Can you spell out for us what the major takeaways were from that discussion?
Yeah, thanks, Stephen. Tony Wood and I discussed his vision for GSK's pipeline as the pharma is going into this phase of transition. So at the start of the new year, we're looking at a new CEO at GSK. So overall, the message from him was that he expects things to be relatively consistent. Luke Mills is the new CEO, is someone that he's been working with for several years. And I think in his view, they've done deals together. They've headed late stage clinical development decisions together. And so his view is that a lot of that will continue. So overall, this is a pipeline that over the last 3 years has become very centered in 4 therapeutic disease areas. There's respiratory, immunology and inflammation, oncology, HIV, and infectious diseases. Some of the interesting points from my conversation with him was that this is the focus area, but he's sort of looking for ways to slightly expand beyond those core areas into diseases that are maybe a little bit adjacent based on common underlying biology. One example that I thought was very interesting was the fibrosis example. GSK has an approved product for COPD, which has sort of a big underlying fibrotic mechanism. A long-acting COPD medication is now in review. And he talked a lot about how this expertise in fibrosis can sort of expand into other indications that also have that underlying biology. So specifically, liver diseases, MASH, something that's exemplified through the Boston Pharma deal that GSK did this year.
Well, so Lauren, does that mean when you're talking about complementary areas, are we going to see GSK? Are they going to be another one getting into obesity, or are they going to stay out of that fray?
So I don't know that I can answer that, but another point that he brought up is that specifically with GLP-1 receptor agonists, this is not an area that's GSK's current core expertise. And he said he doesn't have a lot of interest in being, the fourth or fifth player in the GLP-1 market. That's kind of a trend that we've seen in the last few years with GSK. If you think back to the PD-1 mAbs, this is an area where a lot of pharmas went in really quickly. GSK came in much later and is now sort of looking for specific niche indications within that PD-1 space. Like another mechanism that we're seeing a lot of this year is the PD-1 VEGF bispecific antibodies. Again, that's a place where GSK has not sort of followed the herd. I sense little appetite to do that unless it's, a place that falls very squarely in this like core therapeutic area.
Yeah, I read your piece. It was interesting. I remember him using the word discipline. We're going to be very disciplined about this. Maybe the old GSK wouldn't have, but we are. We have biology we understand really well from our core areas, and we're going to see where that biology is useful. Then like the two of you seem like you digged a little deeper into biology through a discussion on how causal biology has sort of evolved over there. So like, can you tell us a little bit about that?
Sure. So he talked a little bit about how under Hal Barron, the last CSO, GSK sort of incorporated genomics into a lot of the drug discovery work that they did, into a lot of the choosing the indications, the programs, the targets, things like that. And Tony Wood and his team have been building on that focus to create more of a causal biology lens that incorporates the other layers. The spatial transcriptomics and single cell sequencing has become a very important part of what they're working on. I think he said there's no going forward without evidence of causal biology.
So, Lauren, what did he have to say about modalities? Are there any specific areas in which GSK is, putting a lot of their energy, or are they agnostic to modality? What was his focus there?
So he didn't say they're agnostic to modality. GSK is actually sort of ahead of the pack in what we're seeing in cell and gene therapy lately, where we've been seeing a lot of pharmas sort of step back from this space for many different reasons. GSK did that several years ago. But within the new modality space, they've been very focused on RNA-based therapies. I think within the hepatitis B space, so applying it to infectious diseases. He talked about a candidate for MASH that's an RNAi therapy. So, as a lot of companies have sort of come to the conclusion right now, RNA therapies, like oligotherapies, are pretty well validated at this point. They're long acting, which I don't know if that was the original expectation. So it's not like a one and done as you would get from a gene therapy, for example. But a lot of these are lasting 6 months or more. So it's getting to the underlying genetics of a disease rather than targeting the proteins. And you're getting a long effect. So a lot of overlap between the indications that you could address with a gene therapy and an RNA therapy. And that's just where they're putting their effort at this time.
Yeah, I think the way he put it was, quote, "I'm much more interested in things that are readily scalable. Right now we're building a presence in oligonucleotides, in contrast to these other sorts of genetic medicines." there is a scalability element to the RNA side of things.
No, excellent point. Well, great. Well, thank you, Lauren, for that. As I said, it's been available on the website now, so you should go check it out. It's a really interesting read. Now we're going to head to Washington. Steve, couple things I think that we need to discuss, but maybe off the top here, the FDA commissioner has officially launched the first set of national priority vouchers. Can you tell us what's going on here?
Yeah, so the first batch of Commissioner National Priority Review vouchers was a bit, what I'd say a dog's breakfast, right? It was some investigational drugs. It was some drugs that have been approved in other jurisdictions, some generics. So what did we learn from it? I think the most important thing is that this program is a reward. It's not an incentive. It really isn't a voucher. It seems that name was adopted because priority review vouchers are very effective and popular. But unlike PRVs, the commissioner's so-called vouchers can't be transferred or sold. And critically, unlike PRVs, there's really no way to design a development program or change it so a company or an investor can know that it's going to be eligible for this. So instead of saying, well, here's the criteria, for example, first approval of a drug for a rare pediatric condition, and then making it clear that every drug that meets that criteria gets a voucher, instead of that, the new program basically says anything FDA likes, might get one, might not get one. So it really looks like FDA picking winners. And that's certainly how the markets interpret it. Shares in the companies with drugs that were selected jumped. And that's a reasonable bet because the hallmark of the new program is FDA working proactively with companies to get rapid approvals. So the fact that they've gotten one of these vouchers, so-called vouchers, is validating.
What do you think are the chances that they're actually going to be able to hit these targets that they've put out of like 1 to 2 month reviews to get these done? I mean, I guess we have seen other pretty rapid timelines when there's been overwhelming efficacy. I think either the PD-1s or the CAR-T, some of the first CAR-Ts, maybe those were quite rapid, but I don't remember off the top of my head how fast those got done. But 1 to 2 months, in the current environment seems like a tall order. Is that realistic in your mind?
Well, it could be. It depends on when you start the clock ticking, right? And I think that the idea is that these are going to be rolling submissions. They're going to be working with them, the companies, and the clock starts ticking when the final clinical data is in. So it's not really clear whether they're really going to, how much faster they're going to be. Some of the rate limiting work that FDA has to do, especially negotiating labels, for example, really can't be completed until the full clinical data package is submitted. So, that remains to be seen. I think there's another thing, though, that's critical, which is that there's going to be a cost to this. FDA's staffing is down. We ran a chart about that last week showing CDER staffing reductions this year. So if staff is prioritizing these products, it will have less time to devote to other applications. But I think there's another thing to think about in the way that the agency is picking winners here, and in doing so, essentially creates losers. For some types of products, there's a real benefit to being first. We've seen this in gene therapies, where in some instances competing products just drop out after the first product is approved. And there's a lot of reasons for this. One is that for some rare conditions, there's a pool of untreated patients, and then a trickle of new patients after that pool has been treated. If the first product to come to market benefits from meeting that pent-up demand, then there's not going to be that much market left for companies that come in later. And in some cases, if a company jumps the queue and it gets a full approval, then it could preclude another product from receiving accelerated approval, because you can't generally receive accelerated approval for a competing product if it's not an unmet need, if there's something that's been approved for it.
So, Steve, you talked about the pathway lacking predictability, the predictability it would need to be an incentive that can steer behavior toward priorities. But when you look across those first batch of vouchers, do specific priorities sort of jump out at you?
Not really. I mean, and one thing, so the criteria for picking winners here is very vague, it's very broad, and it actually opens FDA to charges of favoritism or even cronyism. I'm sure some people are going to question its decision, for example, to award a voucher to a contract manufacturer, as a contract manufacturer called Flow, that's Flow with a PH, for producing ketamine API. probably most of BioCentury's audience has never heard of Flow. It was in the news in 2020 when it was a startup. The first Trump administration awarded it a $345 million contract during COVID. The company was championed by Peter Navarro, a senior White House advisor. Navarro's back in the White House in the second Trump administration. In 2020, there was criticism of giving such a large contract to a company that had absolutely no track record and a CEO who had been involved in controversies over pricing and manufacturing quality at his previous companies. And it's clear, Flow has retained its connections to the White House and to the Trump administration. It's gotten this voucher, but there isn't a lot of transparency around why ketamine, why Flow, why a contract manufacturer. That wasn't really envisioned when the program was described in the first place.
Steve, do we have any sense as to how many of these vouchers the FDA or the administration intends on issuing?
No idea. So they'd said initially, a few months back, they said, we're only going to give 5 in the first batch, but they gave 9 this time, right? And then when they gave them, they said, stay tuned. Soon we'll have another batch of vouchers. So it's completely unclear. I should say also that, one of the vouchers was given explicitly, the FDA commissioner said it was explicitly given as part of as a reward for most favored nation drug pricing. Another one was given to a generic drug manufacturer as a kind of reward for onshoring manufacturing of an antibiotic, Augmentin. They're being used as tools for various purposes, for various priorities or various policy goals of the administration and of the FDA commissioner. And that's another part of the unpredictability. It's not predictable who can get one, what is it that would enable you to get one, or what would preclude you from getting one, and how many they're going to be. When the program was first announced, the FDA commissioner said that they would only be available to American companies, to U.S. companies. And I actually questioned the HHS press secretary about that, who got back to me and said, of course, they're only going to go to American companies. But that hasn't been the case at all. Several of the companies that have received these have not been U.S.-based companies. And as far as I can tell also, it doesn't seem to be a uniform requirement that the manufacturing of the products take place in the United States. Some of them, they highlighted the fact that the manufacturing is going to be done in the United States, but some of them, it looks to me like that may not be the case.
So, Steve, part of the reason I guess I asked that question about if they have an idea of how many of these they're going to be handing out is because I think it dovetails nicely with the second sort of bit of news that dropped last week about staffing levels at FDA. Because I think in particular, at least to me, it seems like if you're going to be doing these vouchers, you're going to be then sort of a lot of work to get done if you're going to be doing these reviews in 1 to 2 months. And so that could potentially eat up extra resources at FDA. And so I'm just curious, if we can talk a little bit about, what we saw from the staffing report last week and how that maybe plays into what's happening with the vouchers as well.
So staffing fell at CBER by 14% and at CEDAR by 13%. From June 30th to September 30th of this year. I think that we're going to be seeing more reductions ongoing. I keep hearing from people at FDA about their plans to leave. And I think it's critical not only just the numbers, but who's left. A lot of expertise has left. A lot of review staff have left. A lot of the support staff who are needed to make reviews happen in a timely manner have left. So what do they prioritize? When you lose staff, you have to figure out what you're going to prioritize. What I'm hearing is that FDA is prioritizing hitting the numbers, right? Hitting their PDUFA goals. They're going to prioritize, obviously, hitting their Commissioner's National Priority Voucher goals. Some of the things that are likely to slip, and what I'm hearing anecdotally are already slipping, is the kind of advice that companies, especially small biotech companies, need to get from FDA to make their applications kind of flow smoothly. The big companies, have all the resources that they need in staff. They don't lean on FDA as much for advice, but small companies really have come to depend on scientific and regulatory advice from FDA. And I'm hearing that it's getting more difficult for companies to get that advice. It's more difficult for them to arrange in-person or virtual meetings. And in more cases, FDA is insisting on giving answers in writing, which are it's less interactive. There's less nuance to it than what the companies want.
So is the expectation then that, do you think companies will start turning to some of the other regulatory agencies for that to try and supplement with that advice information? I know obviously EMA has, put a lot of effort and time into trying to create these pathways that provide early advice for companies. I'm wondering whether they might start seeing an influx.
I haven't heard that. And I think that there's so many other factors that go into considerations of what jurisdictions companies try to launch their products in and develop their products for. I think that the reimbursement challenges in Europe, both in terms of the amounts that companies get for reimbursement and the timing, the delays in getting reimbursement and things like that, are likely to overwhelm benefits that they might get from getting earlier scientific advice in Europe.
Sure, fair enough. So, the other thing that comes to mind, Steve, now, 14+% in what essentially amounts to 3 months. Is that right? Did I hear you that correctly?
Yeah.
That seems pretty severe, actually. So staffing at FDA was one of the biggest concerns that investors kept raising with me, over the course of this entire year. That was one of their major concerns, worries that they were worried about. And then when we started actually seeing, and hearing about people leaving, that was what they viewed as kind of one of the biggest threats. Is this as bad as they thought it might be? Can we, on a scale of 1 to 10, how bad is this?
I think it's bad. It's probably not evenly distributed. There are probably some divisions or at FDA that have been hit more than others. There's kind of a variance in the level of experience and in the level of expertise of some of the people who have left. The thing I think that's also probably or should be concerning to companies is that we're not seeing a lot of recruitment. It's not like FDA's trying to backfill the positions of the people who have left. And in fact, I think we're likely to see a continuation of this trend of people leaving.
For what it's worth, just because I like to think about it this way, those percentages work out to 1 in 7 CBER employees leaving over that 3-month period, and 1 in 8 at CDER.
And another thing to think about is that when people leave, very often it stresses the ones who have stayed behind because their workload increases.
Change. No,
it increases. In many cases, it increases because they have to pick up the slack for the people who have left. And in some cases, it may be that FDA is just simply not going to do things that it used to do, or it's not going to do them as rapidly. People who pay attention to the FDA website see that there are many things that are not being updated as rapidly or consistently or efficiently as they used to be. And I think that that's the case for other kinds of interactions that companies have, both with the public and with individual companies.
Well, thanks for that, Steve. You can find both those. We've got stories about the FDA commissioners' national priority vouchers, as well as a story about the FDA staffing that is also up on the website. So that is it for us today. Thank you, as always, for joining us. You can obviously tune into our sister podcast, The BioCentury Show, where the most recent episode featured Selena's debut in conversation with Nello Mainolfi, founder, president, and CEO of Chimera Therapeutics. If you like the show, don't forget to subscribe and leave us a comment or review. You can find all of these stories at biocenturypodcast.com. Thanks for tuning in. We will catch you next week.
BioCentury would like to thank Evotec for supporting the BioCentury This Week podcast. To learn more about how Evotec can support your goals through pioneering drug discovery and development solutions, go to evotec.com/innovation.